Welcome to this technical guide on Halo 96 PCA-EQ and other CFU equivalent assays. The colony forming unit or CFU assay has been a mainstay in the hematopoietic stem cell transplantation field since it was adapted for human cells by Pike and Robinson in 1970. Although not required for bone marrow and mobilized peripheral blood transplantation, it is still required as part of the cord blood standards for net cord factor accreditation and the National Marrow Donor Program, NMDP. On September 29, 2013, the 5th edition of net cord fact standards will take effect. At that time, the CFU assay will no longer be a sole requirement. Alternative and validated assays will also be allowed to be used by the cord blood community. Alternative assays to the CFU assay have been allowed by the FDA and AABB for some time. We've developed assays so that similar growth factor cocktail formulations can be used to those presently employed in the CFU assay. This is Ivan Richin. As CEO of Hemogenics, I shall be describing assays that can be your first step towards high quality results for hematopoietic cell therapy processing. Halo 96 PCA-EQ is an instrument-based methylcellulose-free equivalent to the classic CFU assay. It incorporates the same growth factor cocktails as the MethoCult reagents. Halo 96 PCA-EQ is also fully validated and can replace the CFU assay. Other assays developed by Hemogenics for hematopoietic cell therapy applications are shown in this slide. StemPredict is used to assess cord blood bankability and optimize mobilized peripheral blood collections. Halo 96 SPC-QC and its mesenchymal stem cell equivalent luminescent 96 HU-QC is used to ensure stem cell quality during processing and other procedures such as cryopreservation. Halo 96 PQR and its equivalent mesenchymal stem cell assay luminescent 96 PQR are potency, quality, and release assays. These two assays are the only commercially available potency assays on the market that comply with regulatory requirements. Halo 96 PMT are assays to measure time to engraftment and monitor global patient reconstitution. These assays will be discussed in other technical guide videos. It should be emphasized at the outset that Halo 96 PCA-EQ and its sister assays, HemoFLUOR and HemoLIGHT, were originally derived from the methylcellulose CFU assay. It should therefore be expected that correlative results would be obtained. HemoFLUOR 96 PCA-EQ and HemoLIGHT 96 PCA-EQ are recent additions to our product line, since not all labs may have access to a plate luminometer to perform the Halo 96 PCA-EQ. By modifying the readout but maintaining the same cell culture components, we've been able to provide you with high quality assays that can be used in virtually any laboratory. Colony Grow, Cameo 4 and Cameo 96 are all methylcellulose CFU assays. No assay should remain stagnant, and the development from a simple methylcellulose clonal assay to a highly advanced assay such as Halo is all part of providing you with products that give you results you can trust. Let's take a look at some of the advantages of using Halo, HemoFLUOR, and HemoLIGHT Progenitor Cell Equivalent Assays instead of the classic CFU assay. All assays are instrument-based and non-subjective. Low coefficients of variation exist because all assays incorporate the same methylcellulose-free suspension expansion culture technology that produce low pipetting errors compared to MethoCult products. All assays use 96 well plates, and it's not really necessary to use the whole plate at any one time. For all assays, cells can be incubated for 5, 6, or 7 days, depending on your work schedule, without any deterioration in results. In fact, for some assays there is a significant increase in sensitivity from 5 to 7 days. I'll show this later in the presentation. After the incubation time has elapsed, results are ready between 30 minutes and 3 hours, depending on the assay readout. All assays are easy to learn, fast to use, and more cost effective than any CFU assay. There are specific advantages to using HALO 96 PCA-EQ. HALO incorporates an ATP bioluminescence technology that provides the greatest sensitivity of any assay readout. HALO sensitivity is greater than HemoFLUOR, which in turn is greater than HemoLIGHT. Measuring ATP bioluminescence correlates directly with cell proliferation, cell number, and cell viability. The assay includes standards and controls to calibrate and standardize the assay prior to measuring samples. Incorporation of these standards and controls has allowed the assay to be completely validated according to FDA bioanalytical method validation guidelines and allows you to validate the assay in your own lab. Results after cell culture are available in 30 minutes, and a full assay plate readout is available in 5 minutes or less. Based on the growth factor cytokine cocktail used for methylcellulose CFU assays, this table shows the equivalent HALO, HemoFLUOR, or HemoLIGHT comparable instrument-based assays and the type of cell population detected. The most primitive cell population detected is the CFC GEMM3, equivalent to the PCA5 assay or H4434 enriched MethoCult formulation. The Hemogenics PCA5 formulation, however, includes thrombopoietin or TPO and therefore produces megakaryocytes as required by the definition of GEMM, granulocyte, erythroid, macrophage, megakaryocyte. Both CFC GEM stem cell populations are more mature than the CFC GEMM, and the GM-CFC granulocyte macrophage populations are more mature than the others. Now, HALO 96 PCA-EQ, HemoFLUOR 96 PCA-EQ, and HemoLIGHT 96 PCA-EQ are available in 3 sizes, 1, 2, or 4 plate kits. All assays are shipped frozen. Upon arrival, please inspect the box and check the contents. Remove the documentation from the box and store the assay at -20 degrees Celsius until used. Here we have a list of the assay contents for the different types of PCA-EQ assay kits. Always included is a PCA-EQ master mix and medium. For the ATP assays, there's an ATP enumeration reagent, ATP standard, and ATP controls. For the fluorescent assay kits, there's what's called a GL-APC reagent. And for the absorbance kits, there's an MTS reagent. For all kits, there are sterile 96-well plates for cell culture. And for the ATP and fluorescent assays, there are non-sterile 96-well plates for the controls. Also included are sterile adhesive foils to maintain sterility of unused wells, and of course there's an assay manual. Now in this example, 4 cell samples will be tested using the PCA1 master mix formulation. This is similar to the MethoCult H4434 classic formulation and stimulates CFC GEM2 using EPO, GM-CSF, IL-3, and SCF. 0.9 milliliters of the PCA1 master mix will be dispensed into the tubes. The 4 cell samples have been prepared and the cell concentration adjusted to 500,000 cells per milliliter. It's important that when using human cells, the red blood cell concentration is reduced to about 10% or lower. This is because red blood cells can cause false positive results. Once the PCA master mix has been dispensed into all 4 tubes, the cells can be added. It's recommended that the cell concentration be based on the mononuclear cell concentration, not the total nucleated cell count. This is because the TNC contains dead and contaminating cells that dilute the concentration of the proliferating target cells. In addition, the dye exclusion viability should be 85% or higher. Since the PCA master mixes do not contain methylcellulose, you can use normal pipettes to perform all dispensing operations. Just make sure that the pipettes being used are professionally calibrated and, if possible, use electronic pipettes rather than manual pipettes. Pipetting errors will then be reduced to a minimum. When the cell suspension is added to the PCA master mix, the concentration is reduced tenfold. A further tenfold reduction in concentration occurs when 0.1 milliliters of the PCA culture master mix is dispensed into replicate wells of the sterile 96-well plate. Once the cells have been added to the PCA master mix, use a vortex mixer to ensure the contents are mixed properly. The culture master mixes from all 4 tubes have now been dispensed into the 96-well plate. Just replace the lid and transfer the plate to the incubator. The cells are cultured in a fully humidified incubator at 37 degrees Celsius in an atmosphere containing 5% carbon dioxide and 5% oxygen. Culturing hematopoietic cells under low oxygen tension is advantageous because it replaces the production of free oxygen radicals, thereby increasing cell growth and plating efficiency. Now let's take a look at some of the results. This graph shows a comparison of results between Halo 96 PCA-EQ and the Cameo-4 CFU assay, focusing on the MethoCult H4434 formulation using PCA-1 equivalent assay. As an additional comparison, results for the CFC GEMM are also included. The Halo results were performed on day 5 and day 7. Colonies produced in the Cameo-4 assay were counted on day 7 and again on day 10. You will notice that for the Halo assay there is a greater than 3-fold increase in the ATP concentration between day 5 and day 7 of culture for both the PCA-1 H4434 formulation and CFC GEMM. No such change occurs for the colony counts, because once a colony is formed, it only gets larger in size. No new colonies are actually produced. The same experiment performed for umbilical cord blood shows slightly different numbers but the same pattern of results as for bone marrow. You will notice that the coefficients of variation increase slightly for the ATP results as they go from day 5 to day 7. However, this is compensated for by the greater than 3-fold increase in sensitivity of the ATP assay. No such increased sensitivity occurs for Cameo-4 CFU assay, or for any other CFU assay for that matter. These results indicate that you can, without any reservation, replace the CFU assay with the more advanced, standardized and validated Halo assay that is faster to learn, easier to use and cheaper to run than any other CFU assay. Assay results must be meaningful. It's important to emphasize that proliferation comes before any cell differentiation. Samples tested for their functional ability should be tested based on their ability to proliferate. If the cells do not or cannot sustain proliferation, an assay such as the CFU assay will be of no use. For those of you in the cord blood field, please consider changing to the Halo assay. After September 29, 2013, you'll be allowed to do so. But get ahead of the curve and contact us on how we can help you make that change. We look forward to hearing from you and thank you for your attention.